So today we're going to start a new chapter, which is chapter 3, and it's about enzymes. Right? So what are the learning outcomes you'll cover for enzyme? Enzymes are globular protein that catalyze metabolic reactions inside or outside the cells. Number 2, explain the mode of action of enzymes. Number 3, investigate the process of enzyme-catalyzed reactions by measuring the rate of product formation using catalase enzyme and substrate disappearance using amylase enzyme. Now, the 4th one you're not covering today, so we don't have to talk about it now. Okay? Okay, so what is enzyme? If you look at this diagram, here's an example of the structure of an enzyme. It is actually made up of protein, which is globular. So it's a globular protein, right? Because enzyme need to function using its specific shape. So structure function. So using specific shape, it will catalyze specific reaction. So enzyme is a globular protein and it catalyzes metabolic reaction. Now, what is metabolic reactions in our body? Metabolic reactions are all the chemical reactions in the body, which you can represent them using the chemical equation like A plus B to form C plus D. So this is an example of chemical equations. So all the chemical reactions in the body, which you can represent using the chemical equations like this, they are all called metabolic reactions. And all these metabolic reactions must be catalyzed by enzyme. So this word catalyze means what? Catalyze means to speed up. Okay, to speed up. So enzyme catalyzes metabolic reaction means enzyme speeds up all the chemical reactions in the body and remain unchanged after the reaction. So over here, you can see that I'm showing you a polypeptide chain on the left side, which bend and fold into its tertiary structure, which is a precise, compact, globular shape of protein. All right. So in this enzyme, when it bend and fold into this specific tertiary structure, it will function based on its structure. So if I'm to draw the shape of the enzyme like this, okay, an overall 3-dimensional shape, okay, then there's be one location where there's a little pocket where substrate can sit in. So if I draw enzyme, right, I'll draw it this way, right, where over here there's a little pocket where substrate can sit in. So this location where the substrate can sit in is called the active site of the enzyme. Site means location. Active is because when the substrate sit in, reaction will happen here. Something will happen here. That's why it's called active. So active site is the location of enzyme where the substrate will sit in. Okay. So I want you to look at the active site now. When the substrate sits into the active site, keyword, it can sit in, right? So when it sits into the active site, what happens is that it will be held. Okay, my keyword is gone. Sit. It will be held at the active site. Okay, what do you mean by held? Can you see all this R group here of the enzyme that is located at the active site? Now, this R group will actually be holding the substrate in the active site. Hold means what? Remember, when you hold hand means to form bond, right? So hold means to form bond. So which means to say that they will form bond with the substrate. Can you see that? They will form bond with the substrate in order to hold the substrate in the active site. So this is called holding it. Holding it means you hold hand, means you form bond. Now, all these bonds are temporary bond. So it can be hydrophobic interaction. It can be hydrogen bond. Okay. So this is to hold them into place. Right. So it depends on substrate here. If the substrate here is hydrophobic, then this R group is also hydrophobic. They'll form hydrophobic interaction. So the substrate here is positively charged and then this R group is negatively charged, then they will form ionic bond. Okay, so that's how substrate is held. Then we have got this R group, which is triangular in the active site. So the triangular R group is called the catalytic residues. Okay, catalytic residues means they will take part in breaking the bonds of the substrate. So that's why it's catalytic, means they have to catalyze the reaction. So they will take part in breaking the bonds in the substrate so that the reaction will take place. Okay, so that's what you can find in the active site of the enzyme. Right, so remember, sitting and holding are two different things. Right, you sit onto the chair means you just sit onto the chair because you can sit onto it. Hold it means you tie yourself to the chair using a seat belt, for example. So here, the substrate is held to the active site by the R groups by forming temporary bonds. So substrate is held by temporary bonds with the R groups of amino acid residues in the enzyme active site. Okay, now enzymes are named according to the substrate it acts upon. Okay, so substrates are the chemical or the reactants on which the enzyme work. So reactants are chemical that will react. Okay, so all enzymes will end with the suffix ase. And then how do we name it? It's named based on the substrate it will act upon. For example, sucrase will catalyze the breakdown of sucrose, maltase breakdown of maltose, amylase breakdown of amylose, which is starch, urease, urea, and catalase is H2O2. The one in pink color are the ones that you need to memorize. What the reactions they catalyze, which you look at later on. Okay, lipase and protease will catalyze the breakdown of lipid and protein. All right, so you can see that I actually divide the enzymes into two big groups, right? The one on top, all these 5, and the one at the bottom, these 2. Okay, so why is it I divide them into these 2 groups? Can you see or not? Why do you think that I divide into these 2 groups? Recording. Now, when you study lipid, you define lipid as a diverse class of molecules, right? Which is not soluble in water, but soluble in inorganic solvent, isn't it? So which means to say, there are all sorts of molecules that classify under lipids. So how can this lipase enzyme catalyze the breakdown of all different shapes of substrates, you name it? Triglycerides, phospholipids, cholesterol, steroids, all of them are lipids, but they're different shape. Even triglyceride, they may have different shape because the fatty acid tails have got different length, means different number of carbon, and they got different number of double bonds, isn't it? So definitely the shape is going to be different. Proteins also same thing. All sorts of protein, they got different number of amino acid, different sequence of amino acid, different secondary, tertiary, quaternary structure, right? So can you see lipase and protease? This enzyme, they've got more generalized function because they've got many different kind of substrate of different shapes, but all classified under the same group is either lipid or protein. But the sucrase, maltase, amylase, they all only got one specific substrate only. So that's the difference between the 2. Okay. So sucrase, maltase function is more specific, lipase, protease function is more general. So usually we say that enzyme is specific. Now this is a very important word that you need to remember. Enzyme is said to be specific. So what do you mean by specific? Specific means one enzyme can only bind to one type of substrate molecule. So this is the meaning of specific. Okay. So why is it so? It's because the enzyme active site shape is complementary to the shape of the enzyme. So just now I draw already, if this is the enzyme, okay, the substrate must be this shape. So it is complementary. So that means when this part of the enzyme curve inwards, this part of the substrate actually jut outwards so that both of them can fit with each other like the lock and key. So this is called complementary. Complementary means when one side jut outward, the other side have to go inwards. So this is called complementary. So my question now is complementary considered as a same shape, Hoshini?
No.
No, complementary is not same shape, right? If same shape means like that, I got this shape which is like this, okay? And then I got this shape which is like this. And then can we fit to each other? We cannot, isn't it? Because both are same shape. I turn around this way also cannot fit, correct or not? So when you say same shape and same shape cannot fit. So it must be complementary shape, means lock and key. Okay, so this is very important for you to remember. Complementary shape is not same shape. Okay, so let us look at only substrate with the complementary shape to enzyme active site can sit into the enzyme active site, like this one here. This one cannot, not complementary. This one, not complementary. This one, yes, complementary. Then reaction happens. Substrate broken down to products. Okay. So two ways enzyme can fit into the enzyme. Fit means sit into. So please learn up these two words. Sits. So fit into means it can sit into. So first way is lock and key hypothesis, where the shape of the substrate is like the key. It is complementary to the shape of enzyme, which is the lock. So lock and key hypothesis. So this is applicable for all the 5 enzymes that I showed you just now that are specific in their function, specific in their substrate. So what about those 2 enzymes that are more general? How can it be that the different substrate, right, they can also catalyze the reaction? It's because how the substrate sit into the enzyme active site is by induced fit hypothesis. Induced fit. So what do you mean by induce? Okay, so when you go to shopping mall nowadays, you got the glass door that's sliding, right? And then sometimes they don't open automatically one. You have to go and press a bar at the center of the glass door, then only they'll open, like Sunway pyramids like this. You have to press the bar. And then some of the Family Mart also have to press the bar for the glass door to open, right? So when you go and press the door, you are inducing the door to open. Okay. So in the same way, when the substrate, where its shape is actually not complementary to the shape of active site, go and touches the active site will then mold around the substrate. Can you see? The original shape here is not complementary to the shape of the substrate, but when the substrate sits to the active site, the enzyme will be induced to mold around the substrate. And now you can see that the shape is exactly complementary between the active site and the substrate. So that is how the lipase and protease can catalyze the hydrolysis of lipid and protein by this process called induced fit. Okay, the shape of the lipids are slightly different for different lipids, slightly different. So that's why when they sit in the active site, the enzyme can mold around it and then it can catalyze a reaction. Okay, so this for the enzyme where the function is more general, like lipase and protease, which mentioned just now. Okay. So substrate will come and sit to active site and reaction will take place and converted to product. So this is how enzyme catalyze the reaction. And you can see that the enzyme remain unchanged after the reaction, isn't it? And it can be used again and again, right? Okay. So enzyme remains unchanged after the reaction. So it can be reused again and again. So only small quantity can is produced in our body. Okay. Okay, so this in this slide is I'm going to show the mode of action of enzyme. Mode of action of enzyme means the steps that are involved for the enzyme to catalyze a reaction. All the steps involved for an enzyme to catalyze a reaction is called the mode of action of enzyme. Okay, so you need to write and draw this into your notes, everything. And then you pay attention to the color that I use. So if you can also use color. Blue color is in word form. All right. Black color is for the enzyme. Red color is for the substrate. Okay. And the labeling is in green color. So when you use color, you can visualize things. You can see it very clearly. Different color mean different things. So you can focus on the details very, very fast and very clearly. Okay. So what is the mode of action of enzyme? Step number 1, enzyme must plus substrate. And then the substrate will sit in already to become the enzyme substrate complex. Okay. So enzyme plus substrate to form enzyme substrate complex. That means the substrate come to sit into active site of enzyme. So after that, in this reaction, because this reaction is A plus B to become C, you got another substrate, which is B. So your first substrate is substrate A. Then the second substrate is substrate B. Because this reaction is A plus B to form C. So you got a second substrate will have to sit into the enzyme active site. Okay, so when it sit in, now you got the A and the B that is side by side already. Okay, so therefore, they will then hold hand and form bond together to form the product, which is C. So A plus B is side by side, they form bond to form the product C. So therefore, this is called the enzyme product complex. Okay, enzyme product complex. So ES, which is this one here, plus another substrate, which is this one here, it will form the EP, enzyme product complex, which is this one here. Okay, so then the next step is the product will then come out from the enzyme. So from enzyme product complex, you get the enzyme back and the product C, which is formed. Okay, so therefore, this is the mode of action of enzyme. So again, E plus S will form ES complex. And then ES complex will then become EP complex. EP complex will form E and P. So this is the mode of action of enzyme in detail. Okay. Whether you have the second substrate or not, it depends on the type of reaction you are talking about, because some reactions might be A broken down to become B plus C. So then you wouldn't have substrate B already. You'll get your product, which is B plus C. Okay. So you can see that only small quantity of enzyme is produced in the body because the enzyme remain unchanged after the reaction, and therefore we can reuse it again and again. Okay. Enzyme has high turnover rate. What do you mean by turnover rate? Turnover means you start with the enzyme, right? And then it catalyzes a series of reactions. And then you get back your enzyme. And then you can turn it back and use it again. So turnover rate means how fast you can use it again and again. So how fast you can turn over it means how fast you can use it again depends on how fast it can catalyze the reaction, right? How fast it can convert substrate to product. It's called the turnover rate, because turnover means you have to start from the original state, which is no substrate, and after the process, you come back as the original state. Can you see you make one cycle already? So this is called turnover. So in one cycle, one reaction has happened. One reaction means the substrate, one substrate converted to product. Okay, so if you say the turnover reaction is high, the turnover rate is high, that means this turning is very fast. That means the many substrate is converted to product at any one time. Okay, so it's called turnover rate is very fast. That means this this process is very fast so that a lot of substrate can be converted to product at any one time. Okay, let's say in 1 second, in 1 minute. Okay, clear? So this is a definition for substrate. Substrate is a chemical or reactant on which an enzyme works. Enzyme substrate complex is a temporary complex, forms when an enzyme binds to the substrate. So that's why it's called enzyme substrate complex. Every time you use the word complex means you got 2 or more things joined together. Okay. Product is an end material of a chemical reaction or an enzymatic reaction. So these are the definitions of these 3 terms. In biology, definition is very important. So you need to learn up the definition. Okay. Now, enzymes. Enzyme can function either inside the cell or outside the cell. Right? So inside the cell, we call them intracellular enzyme. Outside, we call them extracellular enzymes. Okay. So intracellular enzymes are the enzymes produced by the cell and will be used inside the cell. For example, intracellular enzyme act inside the cell and responsible for catalyzing, means speeding up. Okay, catalyzing the reactions, speed up the reactions that occur in metabolic pathways. So metabolic means metabolism, okay, the reactions. Example, anaerobic respiration enzymes, they are found in cytoplasm. Aerobic respiration enzymes, they are found in mitochondria. Photosynthetic enzymes are found in chloroplast. So whenever a reaction occurs in certain location of the cell, is because the enzymes for the reactions are produced in that location. That's why the reaction occurs in that location. So why is it aerobic respiration occurs in mitochondria? It's because those aerobic respiration enzymes are produced in the mitochondrion. So therefore, the aerobic respiration occurs in the mitochondrion. Okay. Lysosome contains hydrolytic enzymes, so it catalyzes the digestion of pathogens and many other materials in the phagosome, autophagy, and autolysis process. Okay. Now we also got extracellular enzymes, enzyme that's secreted by the cell. Secreted means secrete out of the cell. Okay. When you say secrete means the cell will release useful substances out of the cell. It's called secretion. Okay. Now there's another term to describe when the cell release unwanted material. Unwanted material out of the cell. So when the cell release unwanted material, we do not say secrete already. What is the term that we use when we describe the cell release unwanted material out of the cell? Filbert, what is the term?
Unwanted material.
Yeah. Do you remember? Anyone remember?
Remember, actually.
Okay, it's called excretion.
Right.
Remember, right? Remember,
Yeah.
right? Okay, so you see, both secretion and excretion, the cell release chemicals or release substances out of the cell. But depending on the process, the name is different one. If the cell release something that's useful for the cell and useful for the body, we call that process secretion. But when the cell release something that is unwanted materials, we call that process excretion. For example, after phagocytosis, and then we got the lysosome to go and digest the bacteria. And after that, the unwanted material released out of the cell. So that process is called excretion. Okay. But now if you release enzyme out of the cell because you want to use the enzyme to digest things, or you release hormone, okay, because you want to regulate homeostasis, or you release antibody because you want to fight with the bacteria. So all this release of useful chemicals, they are all called secretion. Okay, all right. So this is a difference between the term secretion and excretion. So you need to pay attention to what is the word that's being used for different processes. Okay. So enzyme that is secreted by a cell function outside the cell. So what are those examples that you have learned? Like amylase enzyme, right? They're secreted in our saliva. So they're used to digest the starch. So the starch will be hydrolyzed to become maltose. All right, catalyzed by the enzyme amylase. So we also have got thrombin, right, which is a protease, will secreted by the cell into the blood plasma in order to convert fibrinogen to fibrin in order to trap red blood cells and platelets so that the blood will clot. Okay, so this is another example. Acrosomal enzymes are enzymes secreted by acrosome. Remember, acrosome is a lysosome at the head of the sperm, which is being released so that, you see this is a sperm, right? So at the head of the sperm, you've got this organelle, membrane-bound organelle like our lysosome. So it will actually release. So when it comes near the egg, it will release those enzymes so that those enzymes will digest this layer of cells surrounding the egg. So this is the egg, okay? And this is actually the layer of cells, which we call them follicular cells. So it will release those enzymes to digest a path through this layer of follicular cells in order to reach the egg. So these are all the examples of enzyme released out of the cell. So they're called extracellular enzymes, okay? So this is to show you how enzymes or any other chemicals are released out of the cell, okay? Okay, so now we want to know, we say, okay, enzyme catalyze a reaction, and catalyze means to speed up the reaction. So we want to know how enzyme can speed up a reaction. So there are two reasons why enzyme can speed up a reaction. Two reasons. Number one, because the enzyme reduces the activation energy of the reaction by providing an alternative pathway. Number two, the enzyme holds the substrate into the best position for reaction to occur. Now, I will start with number two first because number two is very easy to understand. Okay, so let's say, for example, we got a reaction whereby A plus B to form C. Okay, so A must react with B to form C. So let's say, for example, the shape of A is like this. Okay, and the shape of B is like this. So now when A reacts with B, C will become this shape. Correct? A bit too long already. Okay, draw again. So C must look like this, right? Where the A combined with B to form C like this, correct? Okay. So imagine A and B are in the solution and they're supposed to react with each other. Now, actually what happened when they react with each other is A and B must come towards each other in this direction and this orientation, and they will collide or hit with each other so strongly. Collision. So they have to collide, means they must hit with each other so strongly that it will become an effective collision. Effective collision means when they collide, the reaction will take place. So A plus B becomes C. So this is the meaning of effective collision. So because why? Because molecule in the beaker of solution, let's say this is a beaker of solution, and you've got a lot of A and B inside it. So you've got a lot of A inside it. This is B. You got a lot of A inside it, and you got a lot of B inside it. Okay, so you got a lot of A. I should use triangle as A, right? And this is B. Okay, so a lot of them inside it. So they'll be moving around. Okay, because of the heat energy from surrounding, you got kinetic energy. They're moving around. Okay. Oh, this is not a good example. Okay, let me do it this way. Okay, let me make B like this. Then the example is good. Okay, let me make B like this. Okay. B is like this. Okay, so at any one time they move around and by chance they will collide with each other. Okay, so what happened is that when the B here collide with the A here, can you see that reaction will not happen one? Do you know why reaction will not happen? Because they are not in the correct orientation. Because when A collide with B, what happened is that this part of B must actually collide with this part of A head on in this orientation. Then only they will hold hand because their hands are here. Their hands are not everywhere else. Can you see that? So what is the chances of them when they move inside this solution that when they collide with each other, the orientation is correct so that they can hold hand and therefore they become C. The chances is very slim, right? Because it depends on chances. When I come to you, doesn't mean that my hand will be facing you and your hand will be facing me, will be holding hand, isn't it? Okay, so therefore the chances is very slim, right? So therefore, enzyme will help them to hold hand more easily because the enzyme, okay, let's say for example, I've got an enzyme. I'll draw it here so that you can see. I erase this away. Okay, so I got my enzyme in blue color. Okay, so my enzyme will have got this shape. Okay, so that when A sit into the enzyme, it will sit in this orientation. And then when B sit into the enzyme, it will sit in this orientation. So therefore, when A and B sit into the enzyme straight away, they are facing each other in the correct orientation, right? Where they can straight away hold hand already. So therefore, you can see what enzyme is doing here is enzyme is actually holding the substrates in the best position for reaction to occur. Reaction here means that A will form bond with B. So the reaction here is we want A to form bond with B. So you can see enzyme holds A and B in the correct position or orientation so that very easily the reaction will take place. So you don't have to keep on turn around, try and error, and then finally your hand can hold each other. So that's why it's very slow because you have to, by chance, keep on trying. But here, no need. Just sit in, can hold hand already. So that's why very fast. Okay, so this is the first reason why enzymes speed up the rate of reaction is because enzyme hold the substrates to the best position for reaction to take place. Okay, now second reason how the enzyme speed up reaction is it reduces the activation energy by providing an alternative pathway. So for this one, to make it easy, we will see the drawing here first. Okay, we don't see this one. Great. So I will use this to illustrate the enzyme catalyzed reaction. So let's say, for example, my substrates are the rock here. Okay, the rocks here. Very huge piece of rock, as big as half your size. Okay, as big as half your size. So you want to catalyze this reaction. So what is the reaction you want to do here is that you want this rock, you want to use this rock to hit the heart, which is at the foot of the hill. But you're in this location now. You want to take this rock to go and hit this heart at the foot of the hill. So what you need to do is you actually need to push this rock up the hill, isn't it? So you need energy to push this rock up to the hill, up to this point. So that at this point, you just have to use your finger to push the rock and the rock will actually slide down the hill on its own. And then finally, it will hit this heart here, correct? So in order for this reaction to take place, you have to first pump in the energy to the rock by pushing the rock up to the hill. And after that, the rock already acquired the energy, potential energy. So therefore, when you push it slightly, it will roll down the hill and hit the heart. Correct or not? Okay, so in order for reaction to happen, you must first pump in the energy. All right. So now there's an enzyme that comes to you to say that, hello, I can help you so that the reaction will take place faster. So how does the enzyme help you now? The enzyme will chop off the heel. Okay, it will chop off the heel and make the heel lower. All right. So you can see that when the heel is lower, now the distance that you have to push to the top of the heel becomes shorter, right? So you have to push the rock up to here now at this point, no longer up to there at this point. So can you see you need less energy now to push the rock to the heel because the heel becomes lower already because of the presence of enzyme. So can you see the enzyme now provide you with an alternative pathway, okay? Alternative pathway. So now it becomes easier, right? You need less energy to bring the rock to the heel. And after that, you just have to push it further. The rock will roll down and hit the heart. So that's what the enzyme do. It makes the heel lower. So you need less energy to push the substrate up the heel before the reaction can take place. Okay, so let's look at in a diagram, in a graph. Just now, I only illustrate now the actual story. So whenever you see a graph, you must first look at what is the y-axis and what is the x-axis. So the y-axis is the energy level, okay, the energy level of the chemicals involved in the reaction, and the x-axis is the time, okay, the time. So we start with the substrate having this amount of energy. So this is the energy level of my substrate at the beginning. All right. And then in order for the reaction to take place, the substrate energy must be raised up to this level. Then only the reaction will take place. Okay, just imagine reaction take place means you have to roll down the hill, but the hill is so high. So you have to pump the energy up. You have to pump this amount of energy, okay, from here, right, the original. You have to pump this amount of energy into the substrate so that reaction can take place. So this is called the activation energy. So the energy that you have to add into the substrate in order to activate the reaction to take place. So activation energy is this amount. Okay? Now, why this amount? Okay, why this amount? Remember just now we said molecules moving in the solution can collide with each other. Okay, so molecules that's moving in the solution, they've got their own kinetic energy and they may collide with each other. So sometimes when their energy is not enough, even though they collide with each other, they're only touching or brushing each other. So when they touch or brush each other, because the kinetic energy is not enough, that collision is called ineffective collision. That means even though they collide, they cannot hold hand, so therefore no reaction take place. Okay, so if you want the collision to be effective so that they be converted to product, both of them must have enough kinetic energy. That means the kinetic energy must be above certain level so that when they collide, it's called the effective collision. So because of that, we must raise the energy of the enzyme up to this minimum level, which we call them the activation energy. So that when they collide with each other, the collision becomes effective. That means they got enough energy when they collide, so the collision is effective. Therefore, reaction will take place. Okay, so that's why we must raise the energy to at least the activation energy or above that. Okay. So then the reaction will take place like this, and then the product is formed. Okay, so this is my substrate. here. And this is my energy level of substrate at the beginning of reaction. And this is my product. Coordination problem. And this is the energy level. Okay. This is the energy level of my product at the end of the reaction. Okay. So between my substrate and product, which one got more energy? Between my substrate and product, which one got more energy? Januli?
The substrate.
You look at this graph here. You know how to plot the graph, right?
Yeah.
This is 0, isn't it? Correct? This is 0, right? So the number increases as you go up the graph, right? So which number is higher? The number for substrate is higher or the number for product is higher? Generally, number for substrate is higher, right? So which one got more energy, substrate or product?
Substrate.
Substrate, right? Okay, so substrate got more energy than product. So in this reaction, product got less energy. So is the energy from the substrate being released? Can you see from this level, now the product energy is lower than substrate. So this amount of energy here has been released from this reaction. That's why the product got less energy. That means you take the substrate, you take away this energy, you get this product. So this energy is released from the reaction, isn't it? As one of the products of the reaction is energy is being released. So that's why product plus the energy. Can you see that? Okay, so that's how you read whether in a reaction it is exergonic or endergonic, right? Exergonic means release energy. Endergonic means it will take in energy. If take in energy, your product should be here. Right? So from substrate, you have to take this amount of energy in order to form product. That means you have to plus this energy in your equation to form this product. Can you see that? So you got your substrate plus this energy to form the product. Whereas over here, you got substrate to become the product plus the energy. Can you see that? Okay, exergonic and endergonic reaction. Okay, so what happens? So this blue color line indicates the pathway of reaction when there is no enzyme. Okay, but the red color line indicate the pathway of reaction when there is enzyme, because remember, enzyme will chop off the heel and make it lower. So that means the enzyme will reduce the activation energy needed for reaction to take place. That's why the red color line is when enzyme is there. All right. So it will reduce the activation energy that's needed, all right, for the reaction to take place. So therefore, it will take place faster. Okay. So the next diagram is what you need to draw into your notes. Please draw this into your notes as a practice. All right. So why is it enzyme can catalyze a reaction? Means why is it enzyme can speed up the rate of reaction is number one, it will hold the substrate into the right position for reaction to take place more easily, so it's faster. Number two, it will reduce the activation energy. So that means from here, without enzyme, okay, this is the activation energy, this amount here, right, from the original energy of the substrate up to this is called the activation energy. The total energy you need to rise from the original energy level for a reaction is called the activation energy. Okay, so now with the presence of enzyme, your activation energy is actually reduced already. So it reduces the activation energy that's needed for the reaction to take place. Okay, can follow or not? Okay, so here we're going to look at when a reaction takes place, what actually happens. All right, so when a reaction takes place, my substrate will be converted to product. So you can see that substrate will be converted to product, catalyzed by the enzyme. Okay. So when I draw the concentration of substrate and product in the course of the reaction, course of reaction means you follow the timing, okay, of the reaction. You follow the timing of reaction and you find out the change in the concentration. This is how it will change. First, in your beaker, all right, in your beaker, you have got your substrate. Okay, so this is your substrate. Then after that, you add your enzyme into the beaker. So you add a certain volume of enzyme into the beaker. So this is your enzyme. So, and then you mix them. So therefore, now you got a solution, a mixture of enzyme and substrate in your beaker. So this is called the reaction mixture. Remember, this is called the reaction mixture. That means when you mix these two together, reaction will occur. So it's called a reaction mixture. So when you add enzyme to substrate, straight away, the substrate will be able to convert it to product catalyzed by enzyme, right? So therefore, at the beginning of reaction, at time 0, you only got substrate but no product. So therefore, at the beginning, can you see, you got your substrate but 0 product. Can you say 0 product? So when you add enzyme, you start to catalyze the reaction. So your substrate will then be converted to product. So you can see that the substrate concentration starts to drop because it starts to be converted to product. Can you see that? So when it's converted to product, your substrate will disappear and your product will appear. So more and more substrate converted, so substrate concentration continue to drop. And more product form, so the product concentration will start to increase. Can you see that? Start to increase. Okay, increase. And then it will level off horizontally. Substrate concentration also will level off horizontally and remain at this concentration. That means all substrates converted to product, they can no longer change in concentration already. That's because there's a certain maximum or minimum that they will remain as it is. Okay. So remember, the shape of the curve is going down this way and become horizontal, going up, curving, 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 going up is also curving up. Curving up this way. What happened? Cannot draw. Curving up this way. Sometimes like this, this apps cannot draw on. Curving up this way and become horizontal. Okay? So remember the shape. Okay, so over here, based on the learning outcome, you need to be able to describe and also carry out experiment to find out the rate of reaction of these two reactions here. Okay, you need to be able to find out the rate of reaction of these two reactions here in the lab. So how do you do that? Okay, first thing is, so what is the reaction that you want to find out? You need to know the equation. So this is a reaction you want to find out. H2O2 is hydrogen peroxide converted to oxygen and water. And you also need to know what is the enzyme that catalyzes this reaction. The enzyme is called the catalase enzyme. So remember, catalase enzyme catalyzes this reaction. So your substrate is H2O2, your product is oxygen and water. Okay? So when you carry out an experiment in the lab, you want to find out the rate of reaction. First, you need to plot the graph. You need to plot the graph of, this is your product concentration, okay, versus time. So that means to say you need to find out product concentration at different timing in order to plot this graph, right? How do you get this graph like this? You have to find out, okay, what is the product concentration at this time, at this time, at this time, then only you're able to plot this graph, right? So therefore, you have to carry out the experiment. And then at different timing here, can you see now? 30 seconds, 60 seconds, 90 seconds, and so on. What is the concentration of product form here? So the product is oxygen. So you're trying to measure the volume of oxygen released at certain time. so that you jot down the value here. And then from those values at different time, you are able to plot the graph, and therefore you get the shape of the curve like this. Okay, so this is how you get the shape of the curve by conducting the experiment. Okay, so you know that the product concentration increases with time. And then in this equation, can you see every 2 moles of water produced, 1 mole of oxygen is produced. So therefore, you look at the curve for water and oxygen is different. Water is double the amount of oxygen based on the equation. Okay? So where is the rate of reaction? So from here, if I want to find out from the graph, I want to find out the rate of reaction. How do I find out? Now, the formula for the rate of reaction is equals to product, okay, final product concentration minus initial product concentration divided by final time minus initial time. Okay? Does this look familiar to you? My product concentration is the y-axis. My time is the x-axis. So in other words, this formula stands for y2 minus y1 divided by x2 minus x1. Can you see or not? Are you familiar with this equation? What does this equation stand for? Shannon?
Gradient.
Yes, very good. It's a gradient, right? This equation stands for the gradient of the curve. So therefore, if I say I want to find out the rate of reaction, that means I go and find out the gradient of the curve, isn't it? Okay, but I've got a problem here. Is the gradient of the curve the same at every point along the curve? Januli?
No.
No, right? Can you see at every point the gradient is tangent to the curve, right? Can you see it's actually different? So since at different time I've got different gradient, where is it should I measure my rate of reaction? Because rate of reaction actually, can you see it's changing with time? And the gradient is actually decreasing with time. So my rate is actually decreasing with time. So which gradient should I take now? The gradient I should take is the beginning. It must be the initial rate of reaction. So I must take it initial means at the start. Why is it at the start? It's because when you do an experiment, you always write a report, isn't it? And the report, you will write materials and method. And in your method, you actually say that, okay, I'm going to use this concentration of substrate. So if I use this concentration of substrate, I will expect your result will tally with this concentration, right? So whatever result they're going to report, for example, okay, the result is my rate of reaction is equals to how much? Then I will actually check, okay, the rate that you're going to report here must tally with this substrate concentration. And this substrate concentration is the substrate concentration at the start of the experiment, right? Because substrate will be converted to product and its concentration will drop with time. So therefore, when is it that I should measure the rate so that the rate is the most accurate and tally with the concentration is as soon as possible the reaction start, means at the beginning. So therefore, you have to take the rate at the beginning of reaction. So that applies the same thing when you do experiment in the practical exam. When you're asked to measure the rate, you must measure at the beginning. You cannot, never mind, chin chai, I measure anytime also can. Sorry, you measure later, the rate actually dropping already. Then your rate is out of the range, of the correct range to get 1 mark already. Understand? Okay. So with that also, this is product versus time, right? Now this reaction is, you're looking at rate of disappearance of substrate versus time. Okay. In this reaction, we are looking at starch converted to maltose, and we are measuring the rate of disappearance of starch versus time. So with that, you got different time. At different time, you measure the amount of starch by looking at the blue-black color when it reacts with the iodine solution. Okay, with the intensity of blue-black color. All right, so and then you plot the graph. The intensity of the blue-black color means the concentration of substrate versus the time. So you can see that substrate actually concentration decreases with time, and you measure the rate at the beginning here. The rate, okay? Measure the rate of the curve at the beginning here, right? So measure the rate at the beginning. And the rate here is the rate of change of substrate versus change of time, because y-axis is substrate and x-axis is time. Okay, right? So please go back and read the articles I've given to you regarding this experiment and this experiment so that you are familiar with how to measure the rate of reaction in exam. Okay, so that's all for today. Please draw the whatever that I've asked you to draw today.